This video is looking at B1.1, and we're going to focus today on carbohydrates. There's going to be two parts to this, one for carbohydrates and one for lipids. We want to be thinking about ways that variations in form allow diversity of function in carbohydrates and lipids, and also how carbohydrates and lipids compare as energy storage compounds. So the pages we're looking at are 179 to 194 for both sections, and we want to be focusing on form and function. So looking at the structural aspects of both carbohydrates and lipids and what their jobs are due to those structures. So carbon is the building block for carbohydrates, and carbon is the 14th most common element on Earth, but without it, life would not exist at all on Earth. So carbon can make up to 4 covalent bonds with other atoms. Examples that it might connect with would be hydrogen, oxygen, carbon, or nitrogen. And as a byproduct of that, it can make chains of any length. Because of the bond angles that occur when it joins up with different elements, it can cause straight chains, or it can cause a zigzag, but also it can make rings. Macromolecules are very large. Polysaccharide means multiple single units. So a monosaccharide is a single unit of a carbohydrate. A polysaccharide is going to be more than about 7. And macromolecules are going to be big chains of either carbohydrate glucose molecules, for example, or amino acids, which are the building blocks of proteins. And they will have an atomic mass unit of more than 10,000 units. So you have a long chain of elements. So when we're looking at carbohydrates, these here are monosaccharides. So they're a single unit of a carbohydrate. And there's 2 ways that reactions can happen for monosaccharides. They can join together. The monosaccharides can join together, and through a condensation reaction, water is going to be produced, and they're going to join up through this oxygen molecule. This here is called a glycosidic bond. And in this process, ATP is required to make a condensation reaction occur. Alternatively, a disaccharide, which is 2 of these units, 2 monosaccharides, could be broken down, and it's going to use water, and energy is going to be released when this bond is broken. So you could break a disaccharide, or you could break a polysaccharide, poly meaning many, into monosaccharides for energy production. So when we're thinking about the structure of carbohydrates, we have monosaccharides, which are single sugar units, usually 3 to 7 carbons. Examples of this are glucose, galactose, and fructose. So you can see those images right here. Disaccharides, which are 2 monosaccharides linked together. And so 2 glucose molecules is going to make maltose. A fructose and a glucose are going to make sucrose, and a galactose and a glucose will make lactose. So maltose, sucrose, and lactose are examples of disaccharides. And then polysaccharides are very large chain molecules. So examples would be cellulose, glycogen, and starch. So when we are looking, where can I put my face here? When we're looking at polysaccharides, starch, there's 2 versions of starch. There's amylopectin and there's amylose. And so amylopectin has more branching going on than amylose. And amylopectin has alpha 1,4 bonds and alpha 1,6 bonds. So a 1 and a 6 bond. This here, if you look at this monosaccharide right after the oxygen there, this is carbon number 1. And so if carbon number 1 is attached to, this is going to be carbon number 6, is attached to carbon number 6, then that's going to be a 1,6 bond. If carbon number 1 is attached to carbon number 4, which would be over here, then that would be a 1,4 bond. So in the case of amylopectin, it has 1,4 bonds and 1,6 bonds. So it has some straight chains, but also some branching. It's going to be more globular. Amylose only has 1,4 bonds, and that causes it to be a straight chain. And then due to interactions with the groups that stick out from that straight chain, it causes it to coil. Cellulose has beta-1,4 bonds, and so alpha and beta glucose structurally are a little bit different in that their OH groups are positioned differently. So if you look down here, you can see for carbon number 1, beta glucose has the OH group up top, and alpha glucose has the OH group down below. And so that is the difference that's going to between an alpha 1,6 bond and a beta 1,6 bond, or 1,4 bond. Cellulose is going to be a straight chain of about 15,000-ish glucose molecules. And this is going to, straight chains of cellulose in parallel will create microfibrils, which are used for the structural components of plants. Glycogen, and also in case this was not clear, amylose and amylopectin, those are both different kinds of starch. So this is essentially like plant fat. It is a storage molecule that plants create in order to have reserves of energy later on. So for example, a potato is full of starch, and the purpose of a potato is to provide stored energy so that a potato plant can grow from that potato in the springtime. Glycogen is similar to amylopectin, except it's got more branching going on. So this is a picture of glycogen here. And glycogen is stored in the liver of humans and other mammals as an energy source. So when a plant or an animal needs glucose molecules for ATP, then they can pull off the glucose molecules from these branched or straight chains, amylopectin, amylose, or glycogen, to have a hydrolysis reaction that's going to make ATP. For cell membranes have glycoproteins attached to the surface. Usually these are oligosaccharides, which are bigger than disaccharides, but shorter than small polysaccharides. And so these are carbohydrates that are merged with amino acid protein structures. And glycoproteins allow for cell signaling, recognition, and connection. So we looked at these when we were looking at membrane structure. So for example, CAM proteins, or if we are looking at antigens, those would also be glycoproteins. ABO antigens are examples of glycoproteins. So that's going to signal to your body which blood type you belong to. And so if you received A blood, you have A antigens, but you have anti-B antibodies. So what that means is in your blood, you have antibodies that are circulating to say to your body, we don't want any B antigens. And if they find B antigens, they will attach to those red blood cells and rip it apart, as well as signaling to your immune system. So this is why you can't get a B blood transfusion if you are A blood. If you are B blood, you have B antigens, so you have anti-A antibodies, no A antigens in my body. But if you're AB, you have all of the antigens, you have A and B antigens, so you have no antibodies, which means you can get any kind of blood type. You can get O, you can get A, you can get B. If you are O, you have no antigens, so you have no markers. And so you actually have all of the antibodies, which means you have to get O blood because you have no antigens. But this also means that you are the universal donor. So anyone can take your blood because your blood has no antigens. It's traveling incognito, essentially. So to summarize, for carbohydrates, monosaccharides, glucose, galactose, and fructose are one molecule of carbohydrate. Disaccharides, such as maltose, lactose, and galactose, are two monosaccharides joined together. Polysaccharides are long chains of monosaccharides. Examples are starch that have alpha 1,4 bonds and alpha 1,4 and 1,6 bonds, amylose and amylopectin. Glycogen, which has alpha 1,4 and alpha 1,6 bonds. Starch is found in plants. glycogens found in animals, cellulose, which is a structural compound formed by polysaccharides, which has beta-1,4 bonds and forms straight chains, whereas these other ones, amylopectin and glycogen, are branched, and amylose is straight chained but coiled. So condensation and hydrolysis reactions, condensation are going to build molecules, monosaccharides to make disaccharides to make polysaccharides, whereas hydrolysis is going to break down those molecules. So from a polysaccharide, ripping off pieces of glucose to make energy. And then lastly, glycoproteins are attached to the cell membrane to allow for cells to signal to one another and join together. And that is all for this video.